Thanks for joining us for the Open for Neuroscience tutorial on the Common Coordinate Framework. Here's a brief overview of what we're going to cover in this tutorial. I'm going to start by giving you a brief introduction to the Allen Institute and what we do, and then I'm going to hand it over to Lydia Ng, who's going to talk about the development and background of the Common Coordinate Framework. Matham Naimi is then going to give a demonstration of how to access and analyze the CCF, and we'll have time for questions from the audience. If you're watching the recording of this tutorial, you can also post questions in the community forum thread that's linked in the video description. Our presenters today are Lydia Ng, who was involved in the development of the CCF from the very beginning, and we'll talk about how it was made, and Matham Naimi, who's going to talk about how to access and use the CCF. The Allen Institute is a biological sciences nonprofit located in Seattle, Washington, with focused research areas in neuroscience, cell biology, and immunology. The Institute also supports cutting-edge research across the world with the Pulgi Allen Frontiers Group. Across the Institutes, our scientific process is focused on tackling complex, broad, and hard problems in the fundamental sciences. Our approach is guided by three core principles: big, team, and open science, which I'll return to in a moment. And our work generates data, knowledge, and analysis tools, all of which we share publicly. To date, we've collected about 14 petabytes of data at the Allen Institute for Brain Science, which is 2.8 million DVDs worth. The most relevant of our principles today is open science, which is that we share all of our data and tools for you to use in your work. Please check out some of the other webinars in this tutorial series to learn about more of our open resources, as well as the Open for Neuroscience Symposium for more talks about how researchers at and outside of the Allen Institute are using these resources in their work. I'm now going to hand it over to Lydia. And if you have any questions watching the recording of this tutorial, please, again, post them in the community forum linked in the video description.
Atlas is a very unique 3D average template brain, which was constructed from the image data of 1,675 mice, and it has been reconstructed as a 10 micron voxel. And then once we have this sort of beautiful artifact, there was a 3-year process to delineate this by a neuroanatomy team, yielding 43 isocortical areas and its layers, and 329 subcortical areas, ventricular structures, and also many fiber tracts. And everything is open, and you can download and use tools. And a good starting point is atlas.brain-map.org. As I said, it takes lots of people and takes many years. These are all the authors from the paper that we recently published last year. So I just want to do a call out to Quanchen Wang and Songlin Ding, who was our lead anatomist. Yang Li did all the registration to create the average template. Josh, Phil, and Benjamin were part of the annotation team creating those 3D structures. Niall and Matham did a lot of heavy lifting of the informatics. Tim and Brandon and David were part of making sort of web tools for this. And we had a whole army of managers to support them. And also a shout out to Julie Harris, who was a co-lead on this project to help wrangle everything together. Okay, so let's start at the beginning. Where did all this data come from? 2,000 mice is a lot of mice. So the data came from our Allen Mouse Connectivity Atlas. It is a rich data set with about projection information on the mouse brain. What is special to us as a person who has to deal with image processing is the way that the data was generated. Instead of the traditional method in which you take a mouse brain, you cut it up and you put it on a slide, a serial 2-photon tomography system keeps the brain intact while it's being imaged. So the brain is in a holder. To take one image, the system actually images a little bit under the surface of the brain it sees, then it cuts, and then it images again. So as a result, we get this beautiful 3D image set that's already got 3D coherency by the time we get it. And then so that was the basis of getting a lot of 3D data for us to build our framework. So having all of these different specimens, now we need to build an average. So building the average is an iterative process. You just don't take the brains and make the average all at once. You got to do it iteratively, and each time you do it, the average that you get gets sharper and sharper. So up along the top row there shows several iterations of this. We started off with our existing Allen reference atlas as the initial template. So it gives us sort of a pose, an angle, similar to the atlas we've been using for a very long time. And all the data gets registered to that. And then we compute an average. This first average you see is kind of fuzzy, but that's okay. And then we do the formal registration. So you also average the deformation field, invert it, and then apply it to average. And then you keep doing this until you reach some sort of convergence. The reason why we do the inverting the deformation field as well is that will give us sort of an unbiased shape, that you don't lock into one corner of the shape because there was an artifact in one of your specimens. So you start off with something that's fuzzy, and over the iterations, it gets clearer and clearer until you get to convergence, which is H on our screen, in which you can see high level of details. And in that box there, we're seeing the somatosensory cortex, and each of those little blobs there are the barrels from the barrel field. There are a tremendous amount of detailed anatomical information in the CCF. You can see that at a gross level simply by doing a maximum intensity projection like you see in the middle row. And then as you dive down deep into each of the images, you get more and more detail. I'll go through a couple examples later on in this talk. So now that we have this beautiful 3D space, the hard job comes to annotate it by our team. So for every structure, it all starts with doing some research about that structure. The team will look at what is currently known about that structure. What has come up, some of the current atlases draw and say about the structure. Then it comes to what can we see about the structure in the average template, and then a set of reference data is also used to inform the drawing. This reference data includes histochemical information, NISL, HCHE. We leveraged the gene expression atlas as well. We leveraged the connectivity atlas and a whole set of transgenic mice. Our lead and senior anatomist would then do some delineation on the average template, doing a sparse annotation just to make sure that the main boundaries are there. And then our drawing team will come in and fill the structure out in a 3D way. This is very time consuming, and at every step you want to make sure you get 3D coherency. Single nucleus or groups of nucleus are then done individually. As the last step, everything is then put together, and any overlaps and inconsistencies are then fixed, yielding then the 3D structure that you see on our website. For the cortex, because we sort of had prior experience of doing annotating cortical structure, we wanted to do something that would obey the sort of the structure of the cortex. Everyone knows that the cortex it's a sheet, and it's curved both anterior posteriorly and also mediolaterally, but it has this columnar architecture. You can't see it in the CCF, but we know it's there. And because all our data, you can see it in terms of, in our single cell data, you can see the apical dendrites and the processes following that columnar organization. So we wanted to build this as part of the framework. So what we did was first we identified where the isocortex starts and ends using the average template and also some Cree lines. And then we using the pia surface and the white matter surface as an initialization, we solve a Laplacian equation to generate all of these sort of equal potential or equal distance surfaces between those two main surfaces. And then from that, we then, for every point on the surface, we did a steepest descent path from the pia to the white matter. And these streamlines would form what we call approximate columns. So F here, you can see a rendering of a subsample of all of these streamlines that is now obeying the shape of the cortex. And then so what do we do that once we have this? Well, first thing it does is allow us to then integrate information from any sort of layer of the cortex to the surface. So then our difficult 3D annotation problem becomes a 2D annotation problem. So in the top row there, in G, you can see just the integration of the average template onto the surface. Already you can see that the average template itself has a lot of information about the primary areas of the cortex. You will see that unique sort of a triangular, well, quadrilateral shape of the primary visual cortex. You will see this this other bright part, which represents the auditory cortex, and then all the barrels and other domains of the somatosensory cortex. As well as the average template, we're able to then project Cree line data onto the surface, projection data on the surface, integrate multiple projection data together to provide some sort of visual topic map. And all of this information is again used by our anatomists to annotate our 2D surface map. And because we have those streamlined, we can then project that back in 3D such that we have a 3D annotation now that is consistent with all of this reference data and also consistent with the curved shape of the cortex. With that, you can flatten the cortex in whatever way you choose. Down below is us taking a totally flattened view, while the top view just looks at what does the cortex look like if you only look from the top down. And the bottom, we flatten it out so that you can see the edges as well. For example, you can see the rest of the somatosensory domains in the images below. With the streamline, you can also do some computation. In A here, down the bottom row, shows that for every one of the streamline, which direction is it facing? Is it facing forward, anteriorly, or is it facing backward, posteriorly, or side to side? We can measure the thickness of the cortex, and we can also measure how upright that streamline is in its current position. Having this now both a cortical, a bendable cortical sheet, now that we're able to model it, we want to know, well, how good is it, how does that represent, or how does that compare to real data? So we, again, from the projection data, we're able to compare the streamline with some thick tufted layer 5 neurons from the SIM1 Cree KJ18 Cree line. The middle panel here with the little purple star shows that where the soma of those infected cells are. And you can see that it's not, you can't see, to see the apical dendrite from those cells, you have to go anteriorly so that you can see the starting bits of the apical dendrite, and to see its projection down to the white matter surface, then you go posteriorly to see that. So then you can see this in K, which is sort of a sagittal cut of this data, that these cells curve, bent forward in a forward way. So if we unbend them, so this diagram shows now, if we took our flat map and if we unbend the cortex by those streamline, what would the data look like? So O, medial-lateral, is now our new coronal view, our flattened coronal view, and P, going anterior posteriorly, is now our new flattened sagittal view. So that we can see when we flatten it out, we can now see that all the apical dendrites then all vertical, and then well, mostly vertical, and then it projects its projection to the white matter lines up approximately in both both our new coronal and our new sagittal domain. We looked at cells from a visual area further in the brain. Again, it confirms that the flattening, we're able to keep the neurons in apical dendrite in an upright manner. Then we look at parts that are a little bit more messy up in the front of the brain when the curve is bent over. So here, when you look at the actual raw data, you can see that the cells are actually horizontal. So the apical dendrite is actually running anterior posteriorly. So with this, after the flattening, you can actually see that we're able to get the apical dendrite upright in our new sort of unbent coordinate system. So we're quite happy with that. And then so we're able to now use this for data visualization and annotations. Okay, now having created an average template, have done all the work in annotation, created a new cortical coordinate system, we can now use the CCF as the backbone that drives our Allen Connectivity Atlas, the website, and also its informatics. Mathan will probably show you some of the data, how you to fetch some of the data from the web app. But if you go into the Connectivity Atlas, the first thing you see is this map, and each of those dots represent a single experiment that was segmented and registered into the CCF. So because everything is all registered, we can actually show the injection site into one map. When you click on it, we'll know what data set it'll come from. And then all our, on the top here, we show the primary images, but what you see at the bottom here are always registered images so that you can compare one to another. For each data set, then the annotation is used to quantify the projection information, both left and right hemisphere. If you are interested in comparing multiple data sets, you can put them all in a cart and put them in a single viewer. And because everything is registered, you can move around in one image, click sync, and the rest of the data will then sync to its nearest location using our registration results. And so then we're basically using our CCF as a giant sort of navigation system in our Connectivity Atlas. Last but not least, we do a segmentation of the projection data and sort of generate these sort of virtual tractography lines. And then all of that can be visualized in the CCF within the Allen Brain Explorer. In Mathan's part of the talk, he will talk about how you can use the CCF and also how to get some of these quantified data so that you can do your own analysis with this, with the Connectivity Atlas quantified information. All right, so we also pushed the cortical view back in the Atlas as well. We know that our team and a lot of researchers are specifically interested in the isocortex, and then that connections and interaction between cortical areas sort of happens anterior, posteriorly, and middle laterally. So having a top view of that data is very helpful to help interpret the high resolution coronal data. Here we show an example of an injection into the barrel field of the somatosensory cortex. Here you can see the sort of very classic somotor circuit going through the primary motor, the secondary motor, and into the secondary sensory areas. And you can also observe the contralateral projection to the other side, and also focus in the somatosensory area, and very well target back to the barrel fill on the other side. Well, connecting the CCF to our atlas as this giant global visualization system is one use of the CCF. The CCF is also great just to look at anatomy. At the beginning of the talk, I showed that, hey, we see these barrels on the somatosensory cortex, and then this is a zoom-in view of these barrelets that you can see in the medulla, and it again, and these barreloids in the thalamus. This is, typically if you're looking, if you're interested in the circuit, you get these schematics that shows, okay, these barrel fill exist, but in order to see them historically, you would have to cut and resection data at those angles. Because of the super resolution effect, some of that is already readily visible in the average template, and that this sort of shows you that there is a lot in the CCF that is still to be mined, and you're able to pinpoint basically which sort of rows of the barrels that your current data set is being observed in your current data set. At some point, I was very interested, while you can see the barrel fill, be interesting to basically replicate some of the schematics that you see in the paper. Here was in terms of just showing what you can do with visualization with the connectivity data. This was simply taking sort of 3 injections in the in SSP, one in the sort of main barrel fill, one in sort of the nose area, and one in the lower jaw. So there is a anterior to posterior sort of organization to these three injections, and as the projection goes to the barreloids in the VP, that topography is flipped, and as it goes further back to the barrelet in the PSV, it is then flipped back, and again you'll see that in the barrelet as well. And because we selected sort of very sort of specific fields in SSP, you can also see how this maps into different visible domains in the rest of the brain, and the ability then now for an anatomist and the research community to start to link all of this data together and have a good anatomical framework in order to sort of pinpoint the anatomy. Okay, so somatosensory is well known. You can see the barrel. And then I got interested, well, what about other systems? What can we see in CCF for other systems? One of the things I was looking at, well, what can we see about the auditory system in the CCF? Here are some of the diagrams of the auditory system that I found by Googling. So from your ear, there's the cochlea, and its pathway is through the auditory nerve, goes to the cochlear nucleus, and then it goes to the superior olive, and then over to the sort of the medial nucleus, of the trapezoidal body. And then, so this is a nice system diagram people draw, and it sort of reoccurs in multiple species. For the cats, they can actually see these shapes, but then for humans and for macaque, these sort of different shapes changes. And for the mouse, typically you will just see these things as a blob. You don't see sort of the crisp shapes you will see in the cat. With the CCF, can we see more than blobs? Looking at other papers, there is potentially the S shape of the lateral superior olive that exists in the mouse, but it's really hard to see. So what can we see in the CCF? So in the CCF, you can actually start to see this little S shape. I don't know if you can see my mouse cursor. And then, and also the trapezoidal body. But what was really fascinating that instead of just taking a coronal view, we were able to take a horizontal view, like look at it upside down. And then when you're looking at upside down, you can see very clear boundaries between these regions. So if we were going to if someone was going to take a task of refining the boundaries between these structures and substructures, there's information in the CCF to do so. So that was just my little segue of the CCF allows me the ability or anybody to go and basically look at anatomy in a little bit more detail, and then also and then correspond and look at reference data. Okay, so we have said that the Allen, we have mapped all the data from our connectivity atlas to the CCF. What other, so our cell types data is also mapped to the CCF, although this is no longer sort of image processing. A lot of sort of manual pinning of sort of small regions of interest is used in order to pin sort of cell types data to the CCF. Many of our experiments from the Allen Brain Observatory that you've also heard in this symposium is also registered to CCF. Here, we have to take a top view of the data, and then we registered top view of the calcium imaging data, and then match that up to the sort of the blood vessels on the top of a 3D volume that's collected of the brain after the experiment. Neuropixel data is in registered CCF. To do that, they do a optical tomography system in order to collect information about the brain and also location of that probe. And they were able to use this data map at the CCF as well. We retrospectively put all our gene expression data back in the CCF, and we're currently working with the Brain Initiative Cell Sensors Network to map all their data in the CCF so that it can be used as an integration point for all of this data. The question we always get from everybody is, how do I map my own data to the CCF? As you heard, the way in the CCF is highly dependent on your data, and that we had to have lots of people specifically work out the mechanism of registration. However, there are many groups that are working on tools as well. I'm going to call out a few of them here, and then we have some links to some other resources that we know of for registration. The first two, Elastic and Ants, for your programmers. So these are basically registration packages that are deformable registration. And the name of the game is that there's a lot of parameters that you need to tune. But we know that in a lot of external research groups that have mapped their whole brain imaging data into CCF uses Elastic and Ants. cell locator is something that was developed for the BI Season Consortium, which allow us to, it's a UI tool that allows us to pin regions of interest in the CCF. A user goes in, look at the CCF, can cut it in any different way that they want that matches their sort of data. And then they can draw the boundary of I took data from here and then produces a JSON file with that information. And we need that information in order to integrate the data from the single cell data from many different groups. Here is a slice reg software. It's mainly dedicated to trying to map single fluorescent images into the CCF as well. So here are some links for you to have a look to see if there is already a solution for the type of data that you have. And I believe our comms team will be posting these links out as part of this so that people can follow it as well. Otherwise, check out our forum, and then you can search for some of the links in our forum as well. So with this overview done, I am now going to hand it over to Mason as he sort of goes through how you can practically use some of the CCF related tools.
Okay, hi, I'm Mason Naimi. Thank you, Lydia, for the introduction to the CCF. I am going to now share my screen for the next part of the tutorial. Okay, do you have my screen? Yes.
Yes.
Okay, so I'm going to go over how to work with the Common Coordinate Framework, which is going to be this next part of the tutorial. Basically, I'm going to cover two methods of interacting with the CCF version 3, or the Common Coordinate Framework. One is through ITK-SNAP, which is a free open source application that lets you visualize image volumes. This is really beneficial for any kind of visualization. If you're trying to look through the data and get a sense for where there's signal, this is a very efficient and easy way to look at that and get a sense of the different features of the data from different orientations. So it's very easy to navigate the 3D space through ITK-SNAP. And then after I go over that, I'm going to show you how to access the Allen SDK and how to access the CCF through that in a Jupyter Notebook using Python. And this is really beneficial for any kind of numerical analysis that you may want to do. And you can also query any published data sets that are there. So the CCF through ITK-SNAP, you can get the instructions for how to operate ITK-SNAP for CCF purposes through this link here. And this will give you there's a Word document that has all of these instructions, but I'm going to go over them in the next slides. So first off, you will want to download and install ITK-SNAP. And if you go to this link, you can install, we recommend installing version 3.6. There are newer versions. I think there's one newer version that is the one they're working on, but this is a stable version that you can use for all of these analyses. And there are also tutorials on just how to use ITK-SNAP at this link as well. And then you can download the relevant CCF files through the link below. And if I go to that link, second, I apologize. Okay, if you click on that link, it'll take you to this directory and you can download the relevant files. So there's a CCF annotation, there's an average template, and then there's these text files that are labels. So real quick, the template, as Lydia described, that's just the average template of the 1,675 mouse brains. The annotation file is the segmentation file. It's each voxel in that space is annotated to an ID belonging to a structure. And these associated label files allow you to identify those structures in ITK-SNAP. So if you were to download these and then open ITK-SNAP, so the next step is for you to open the underlying image. So in this case, we are going to open our average template. But if you had a data volume, you could open that. You can even open multiple images as overlays on top of each other. So we'll start by opening our main image, and we can browse to the location that we've saved this average template. And this is a NRD file format, so that's correct. And we click next, and it gives us some metadata about this. So this is a 25 micron volume that we've downloaded. You can, through our SDK, I will explain that you can download at a 10 micron resolution, 25, 50, and 100. But here we're working with the 25 micron resolution volume. And it automatically has set the orientation as right, anterior, and inferior, which is causing our orientation to be off. So we're going to correct that in the next part. But first, we're also going to load our segmentation. And so if we downloaded our annotation volume, we can also load that in the segmentation part. And just to be clear, for that to download or to load the segmentation, you go to the segmentation menu, click Open Segmentation, and then you open the segmentation file. And again, you click Next, and it loads that file on the same space, and it has to have the same resolution, same orientation. And so when we finish, we see that it's loaded. Now, if we want to fix the orientation, we can go to Tools, and we can go to Reorient Image. And in this case, we want it to be A, so anterior, and then S, superior, and then L for left. So our first axis is the anterior posterior axis, and our origin is at anterior, and then our next axis is the superior inferior one, and our origin is at the superior, and then we're on the left side. So we apply that, and now the orientation is correct, and you can see that we can scroll through the coronal sections, we can scroll through sagittally, and so on. Now, I mentioned the label files, and the label files are important because if you notice, even though we have these segmentations, which, by the way, if you can't see them, there's a scroller down here that lets you adjust the opacity of the segmentation overlay. But if you click on any particular structure, you'll notice that it has some label value, but that label doesn't represent anything right now because ITK-SNAP doesn't know what that's supposed to mean. So what we do is we go and import label descriptions, and those are those text files that were there. And there's two of them. So one is labeled as the ontology color labels. And if you were to open that, then if any of you have been have looked at the Allen Mouse Brain Atlas, you've probably seen a similar color scheme. This is the color pattern used there. It can help if you're trying to create a visual, or if you're trying to look at the structures in a similar color scheme to the atlas that's on our website, which I will go to in a little bit, then you can use this. But as you can see, it can be a little hard to see which structure is these substructures in the same region, which structure you're in. When you click, you of course see the acronym of the structure here, but you cannot tell from the color. So it's helpful to work with the ITK-SNAP labels, which allow you to notice all the structures there. And these colors have been assigned so that they contrast as much as possible with their adjacent structures, so that you could easily notice what structures are there. And similarly, if you were to then load an underlying data set, you could then apply this segmentation on top of that and visualize that. So I'll go through this one more time just to demonstrate that. So here I'm going to open an image, and in this case, I've already downloaded a projection density 25 microns for a specific experiment, and I will show how to do that through the SDK. And if I were to load that, I can see that there is some signal somewhere, but it's hard to tell what the it's hard to tell where any of this is. You can kind of see that this has that same orientation problem that we had. If I open the segmentation on top of this, we now see, and we can adjust the opacity, and we can now see the brain. And of course, we're going to have to reorient the image here. And so now we can adjust the opacity if we want to see the atlas, if we want to see the signal, and we can look at where the signal is, and here it says the structure is in viz-PM layer 2-3, and then we can follow the signal as we scroll sagittally, or if we go to the coronal section, we can follow it, and we can see the signal down here going in the fiber tracks, and we can follow that and see where that leads. And so that's really the value of working in ITK-SNAP, is you can just visually follow through and see where the signal is, and work in all three views and get a sense for how that signal is distributed in the space. And that really is the gist of working with the CCF and ITK-SNAP. It's really useful for this kind of visualization. Now, to get data like that, projection data, to actually do some numerical analysis, and other possible things that you may want to do with this kind of data, you can use the Allen SDK and Python to do that. And there's the tutorial that I'm going to go over, you can access through GitHub. And you can download this and work with that tutorial. So let me go to the tutorial. So the Common Coordinate Framework can be accessed through the Allen SDK. And so the first thing you'd want to do is you want to download, install the Allen SDK. And of course, for that, you're going to want to have Python, preferably Python 3.7 installed, and then you install the Allen SDK using pip. And you can then run Allen SDK in a Jupyter notebook like this. If you want to follow these instructions or see more documentation, you can click on these links, and it'll take you to the Allen SDK page. It has an installation guide and a lot of information documentation for the SDK. So within the SDK, the module that we're interested in is the mouse connectivity cache. This allows us to access the average template, to access the annotations, and to access data or connectivity data. And so these volumes are NumPy arrays, three-dimensional arrays, and the metadata is easy to work with in a pandas data frame. So I like to import those. And then in this case, to visualize, I'm going to use PyPlot if I want to see a section or some part of the data. And the first part is instantiating the mouse connectivity cache. This is, I'm going to refer to it as MCC. This is where I can access the annotation files, the experiments, and so on. By default, it downloads all of that at 25 micron resolution. So you don't actually need to specify 25, but you could specify 10, and then it would download it at 10 micron resolution. You can specify 50 or 100, but we're going to leave it at 25. Also, when it downloads the atlas and it downloads experiments and files associated with that, it creates when you instantiate this the first time, it creates a manifest file in your current working directory, and it creates a directory in there for the atlas, and then it also has a manifest file that specifies where everything is going to go. And so if you are going to create multiple different notebooks or scripts, and you want to access the same types of things, especially the annotation files or the CCF files themselves, it helps to have a central location that you've put all of these things, and then you can just link to that manifest file, and that way you're not downloading the atlas every single time, or the average template, or whatever volumes you've downloaded before over and over again. So in this case, I'm just going to let it create this in this directory. And then once we instantiate the mouse connectivity cache, we want to work with all the data that's in this reference space. And so we want to access the reference space. And the reference space lets us get the annotated volume. So this is basically a 3D array where each voxel inside the space of the mouse brain has been annotated with a specific ID corresponding to a certain structure. And these structures are arranged in some structure tree according to an ontology. And so all of that can be accessed through this reference space. So we get our reference space from the MCC. And if you look at the annotation file in there, you can see that it's shaped just like the file that we had in ITK-SNAP. So if I go to the end, you can see that it's 528, 320 by 456, and that's what we have here. So those are the dimensions. So this is the anterior posterior axis, the dorsal ventral, and then left right, and the units of this are 32-bit unsigned integers. So each value in there is just an ID corresponding to a structure. Because there are different ontologies, different atlases, what we've downloaded here, by default, when we instantiate mouse connectivity cache, it assumes that we want the latest atlas, the CCF v3. And so in our reference space, we tell it to remove any unassigned IDs. So those are IDs that corresponded to previous atlases or previous versions of the CCF. And when we remove those, what we're left with is a structure graph, which we can see here as a data frame that allows us to see each structure's ID, name, acronym, and even its path in the tree where it's associated with the parent structures that it belongs to. At the end, we even have this RGB triplet value that corresponds to the colors in this color scheme here. This structure graph is one way that we can go through our structure tree and look for specific structures. So you could search in here for a structure by name or by acronym and get its ID. Now the reference space also lets us generate these image sections. It takes an orientation value, 0 for coronal, 1 for horizontal, or 2 for sagittal. And then it also takes a position in microns. Here, the volume that we've downloaded is in 25 microns. So each value, each increment is in 25 microns. But the reference space, when it wants to create this image, it takes them in values of microns. So it helps just for readability purposes. I like to create an orientation dictionary, and then this function that converts whatever position I give it in my 25 micron space and converts it to microns for this function. And so I can have it create a coronal image at the 230th position. So 230 times 25 would be in microns, and I can then view this image. And this is actually a good place to also point you to the brainmap.org. So if you go to brainmap.org, you can scroll down and you can go to reference atlases. And in the adult mouse atlas, you can click on coronal sections, and you can see you can navigate through different sections. You can create you can click on different structures in the structure tree in the ontology, and you can highlight those, or you can just click on them here and see what they are. And so if you notice, again, the color scheme here matches what we get from our reference space image generator here. And so this is one way to generate images like this. The other advantage of the reference space, of course, is that it allows us to create masks for specific structures within that space. And that lends itself to any kind of analysis that we want to do with underlying data. If we have some signal somewhere, we probably want to quantify or evaluate where that is and how strong it is in different structures. And so we'd want a way to parcel out each part of that data volume according to these annotations. And so the masks let us do that. The reference space has a make structure mask function. You can give it a list of structure IDs. So from our structure graph here, we can see, for example, that the cortex, the cerebral cortex, has an ID of 688. And so we can put that in there and create a mask for the cortex. And this function, direct only, what this refers to is, this is a structure tree. So you have substructures that belong to a parent structure. In the annotation volume itself, if you were to click on voxels within the cerebral cortex, odds are you won't find a voxel with the label 688 because most of them have been assigned to some substructure, layer 5 of some area in the cortex. And so that will have its own unique identifier. But when you want to access the cerebral cortex, you likely want to access all the voxels that belong to it, its children or its descendants. And so direct only is asking whether you want only the voxels that have the direct label 688. And in this case, you don't. You want all the voxels that exist within the cerebral cortex. So that's false. In some case where there may be a structure and then part of it is assigned to a substructure, but there's a part of it that remains, and you want to see that difference. In that case, then you can set this to true, and you will only get the voxels that are specifically labeled as that ID and no child ID, no descendant. And so when we create this mask, it has the same shape as our volume. We're again working this 25 micron space. The data here is an 8-bit integer, and really the values, if we look at the maximum value there, it has a value of 1. So all the masked values are 1 and everything else is 0. And we can then view the cortex here. We may also want to look at the average template. So this is the template volume that Lydia talked about that was created from averaging 1,675 mouse brains. And if we want to access that, we can get that through the mouse connectivity cache. We can get this template volume. Again, it has the same dimensions here because we've specified that we're in 25 micron resolution. It's in 16-bit integers, and these values, the maximum one is 516, are really just the intensity of the signal in this template volume. And this would be similar to if we had any other data. In this case, this is the average template, but it could be some other data. And again, it would just be some signals in this space that we can then apply the masks to. So when we want to do analysis using the SDK, it really, it's about identifying the structures that we're interested in, maybe using ITK-SNAP to look at where the signal might be, getting a sense of what we want to do, or whatever your particular experiment requires. You identify those. You can create masks for those structures. You can get the coordinates for those masks, and then you can sample your data volume. So in this case, if we want to sample only the cortical voxels of this average template, we can use these coordinates from the cortex mask and pass them to the average template and get slice the average template by that and get the values, the intensity values at each voxel according to that mask. And then we can do our analysis on that. And so the mouse connectivity cache gives you access to all of these tools. It also gives you access to published connectivity data. So you can see what experiments are there. You can run this get experiments function, this experiments method, and get a data frame of these experiments. And you can see that each experiment has an ID. It gives you metadata about the experiment itself, and you can specify if you want to, for example, look at only experiments that have injection in VisP, then you can sample it by that, and you can get these experiments. And then you can pick a particular experimental ID, and you can get its projection density. So this would be the density of the signal in each voxel for that particular experiment. And so you use this get projection density method, and you give it that experiment ID, and it gives you a projection density. And again, all of this is going to be in the same space because you've specified your MCC as that. And so the values in here are floats. They're from 0 to 1, and they represent that projection density in each voxel. We can look in our data here. So this is experiment 5030699254, which is this, and we see that we have coordinates, X, Y, and Z coordinates for where the injection happened. And these are in microns. So since we're working in 25 micron space, we can take the X coordinate, which is the anterior posterior coordinate, divided by 25, and it tells us roughly which section in the 25 micron volume we'd want to go to see where the injection happened. And so if we go to section 348, we can see this signal here that corresponds to where that happened. And of course you can put other values and scroll through just like you would ITK-SNAP, but naturally it's a lot more efficient or just easier to jump around in different views in ITK-SNAP and look at the signal there if you want to do any kind of evaluation of what the data looks like visually. But once we have this data in this space, if we wanted to do that, if we wanted to load this up in ITK-SNAP like I did earlier, this is the same data set that I showed earlier, we can save it. And so I had saved it as projection density 25 with the experiment ID as an NRD file. And that's how I was able to then load that in ITK-SNAP. So now that we have an experiment, if we want to use masks, we can use masks to subdivide this experiment into its various structures. If you look at the number of structures, the number of individual structures that we have in our structure graph, there are 839. And of course, some of these are at different levels of the ontology. So some are parent structures and some are descendants of those. But if you wanted to break down your data into how much signal is in each of these defined structures, then you can loop through all of those, make masks for them. And you can imagine that would be a little computationally intense. So if you have a better sense of the structures that you're interested in analyzing, then that can speed things up. You can specify, for example, that you want to see how this, you want to do some analysis on this signal in the thalamus. And so you search for the thalamus in your structure graph, and you see that it has an ID of 549. And you're interested in all the descendants of that structure, so every substructure in the thalamus. So you can use the reference space structure tree within that and get the descendant IDs for 549. And it gives you a list of IDs starting with 549 itself, and then all the descendants of that. But these numbers are hard to make sense of. So one thing that helps is mapping these to their acronyms, or you can even do their names. So in this case, we want to get these. We want to get the acronyms corresponding to these IDs. Within structure tree, we have an ID acronym map. So this gives us IDs given a certain acronym, and we get that map, and we can reverse it to get acronyms given a set of IDs, and then we can map those IDs that we generated there to their corresponding acronyms, and that would be easier to navigate. And so given these structures, if we want to break down our data and see how much signal we have in each structure, of course, we have to keep in mind that we may be interested in looking at a specific hemisphere. When we make our mask, it generates the mask across the entire space. So if we look at the shape here, the last axis is the left-right one, and it's in 25 microns. It has a length of 456. So the halfway point is that divided by 2, which is how we can split our left and right hemispheres. So we can get all the signal for each structure up to 228, and then we can get all the signal for each structure from 228 onwards, and then we can just get all the signal across the entire space. So that would be for both hemispheres. And when we get that data, we want the sum of the projection density in that structure, for example. And so then we will specify a value of 1 for the left hemisphere and a value of 2 for our right hemisphere. And then we can specify a value of 3 for both the left and right. And if we were to run this, we can then look at the data and we can see that, for each structure ID, there's going to be 3 values, one for the left hemisphere, one for the right, and one that's the sum of the two. And so that's a way to parcel out our data into these different structures for analysis. Now, if you're working with data that you're accessing through the SDK, through the mouse connectivity cache, you don't have to do all of that. It's already been pre-calculated, and so you can get that information through this get experiment structure unionizes. You give it an experiment ID, and it gives you all of this information. So similarly, it has the hemisphere ID, just like we specified. So 1 is left, 2 is right, and 3 is both. And then it gives you the projection density, normalized projection volume, a lot of different values that you might be interested in for a given structure ID. It also has this column, this Boolean column is injection. So this specifies whether that particular record corresponds to an injection voxel. And so you can separate your injection voxels from your non-injection voxels using that column as well. Now, up to this point, I've introduced the annotation volume and talked about how you have these different structures in the ontology, but we've basically worked with the entire structures or structures belonging to a particular parent structure. But one useful thing when you're trying to do analysis with the atlas is to work with structures at different levels. So we've specified these structure sets that are groupings of structures that are go together, that are useful for doing analysis at these different levels. So 2 examples are coarse level structures, which are 12 really high level structures that let you analyze where your signal is kind of across the whole brain. And then there's also 316 mid-level structures, we call the summary structures. So for example, in the coarse level structures, you're talking about, the isocortex, but at the summary level, you now have the individual areas delineated. So you'll have, the different parts of the visual area, the primary visual, and so on. Those will be specified, but you won't have the layers specified. So those would go below the summary level structures. So you get, with the summary level structures, you get higher resolution breakdown of the brain, but not all the way down to the children, the leaf level of the ontology tree. And so to see these different structure sets, you can access them through the ontologies API, and you can get the structure sets and view it as a data frame. And you can search for all the ones where they have the value coarse in the name, because I'm looking for the coarse structure, and you can see that for the mouse coarse structure, it has an ID of 2. So that's the structure set ID. And that allows us to then use the reference space in the structure tree. We can say get structures by set ID, and we can give it the set ID 2 corresponding to all the structures in this set, the mouse coarse set. And it gives us the coarse structures, which are these 12 structures that I mentioned. And so similar to our structure graph, it gives us the same kinds of information, but only for these structures. And we can get those IDs and perform analysis based on those. We can also do the same thing with our summary structure set. We can search for the word summary in our ontologies structure sets, and we can see what summary structures we have. And we're interested in this brain summary structure. There was a mouse connectivity summary structure that was there before, but since then several more structures were added to this summary structure, and so this is the one that we're interested in. And this gives us 316 summary level structures. And we can look at them again, just like in the structure graph, but only for these structures. And then if we wanted to do some analysis with these now, so you're going to do just a quick example, we can get our core structure IDs from our structure graph that we loaded here. And you can see that I have these 12 IDs. And let's say we want to look at experiments that were injected in MOP, then I can go through my experiments data frame and get the IDs for those experiments. And now I have all of my MOP experiments. And then I can get structure unionizes for these experiments, and I give it the experiment IDs. I give it the structure IDs that I'm interested in. So I'm interested just in these course level structures. So I define that, and then I want, let's say, hemispheres 1 and 2, so I can specify what results I want in my unionizes, and then once I get that, I can, if I want to know which side of the brain the injection happened in, let's say I want to do some analysis to compare ipsilateral versus contralateral projections, then I'd want to know where the injection happened. So I can get my unionizes data frame and search for where the is injection column is true. So just to, again, reiterate, so this data frame looks like this. You have experiment IDs, you have hemisphere IDs, and so on, and you have this is injection column. And so I look for the ones that are true, and I see what hemisphere they correspond to, and they correspond to the right hemisphere. And it happens that all of these MOP experiments were in the right. And so I can then perform my analysis based on that. So what I can do if I'm interested in ipsilateral projection densities, I can subsample only the cases in the unionizes where the hemisphere is 2, but I'm interested in projections. I don't want to include the injection voxels, so I'm also interested in where is injection is false. And so this lets me get this subsample, and then I can pivot this table, and I want experiment IDs as the indices and structure IDs as the column, and the values are going to be projection density. And I could do this for other values. You can see that there's other values that you can use for analysis. But here, for the example, we're going to use projection density. And all of this information, again, how exactly these values are calculated, can be accessed through within the SDK documentation. So there's additional information there. So once we generate this projection density table, we have the values, have the structure IDs, have the experiment IDs, and we can basically take each value, take the structures, and apply the acronym ID map to see the structures as acronyms. For better readability, we can get the experiment IDs, and then get the matrix of the values in there and make a heat map using PyPlot. And so then we can see, for example, where for each experiment, how the signal is distributed across these different course level structures. And you can do other types of analyses, for example, where you're comparing structure to structure connections by downloading data corresponding to injections across these different structures, and then comparing those to their projection densities across these different structures. So there's a lot of different analyses that you could do there. Finally, when you look at these numbers and these values, it's hard just looking at the numbers to really have a sense of what that means. Again, ITK-SNAP helps visualize that, but there you're looking at data that was registered. It also helps to be able to go back and look at the raw images themselves. Of course, you can do that again through the brain map data portal. But if you want to do it through the SDK, you can download image sections using the image download API, and you can get the metadata for it through the mouse connectivity API. And so basically you take the ID for the experiment we used earlier, for example, and you instantiate the image download API and mouse connectivity API. You get from the image download API, you pass the ID to section image query and get a sections frame, and this frame links the individual sections. So you see we have 140 individual images corresponding to these sections of the data, and it maps them to their own specific section IDs. And that's really what we want. We want to map the section number to their corresponding IDs. And then we use those IDs to download the images. We also want to get some details, some information on these images. For example, what are the optimal equalization parameters for the intensities in each channel? And so you could get those through the mouse connectivity API. You can get those details, get the equalization values, and create this range corresponding to the minimum and maximum for each channel. Once you have that, you can make a directory for that particular experiment, and within that, if you want to hold these sections, you make a file for that, and then you loop through your section IDs. And for each section ID, you can specify what factor to downsample by. I downsampled by a factor of 4. You can give it your equalization ranges and the file path where you want these images to go, and it will download these images. And so if I go to this directory, I can see that the images were downloaded, and I can open one and I can see the actual signal in that part of the brain. So this basically brings all, I suppose, 3 parts of this data together. You can see the raw images, you can download the values that were, the data volumes that were registered to this CCF and load those in ITK-SNAP. You can visualize them there, and then you can do some numerical analysis using the tools in the Allen SDK. And that concludes the tutorial.
Great. Thank you so much for both Lydia and Maytham. So we've got some time for questions. We've got about 8 minutes. And I see there is a quick question in the Q&A already. Do you happen to have be able to share the Jupyter notebook in some ways such that folks can access it?
Yes. So you can go to this GitHub link at the Allen Institute page, and it's under CCF tutorial. And if you go there, you can download, and this is the Jupyter notebook right here. And you can download that and access everything that was there.
Awesome. Cool. Any other questions? Please post them in the Q&A and we'll take them live. Well, if there are no further questions, I guess that would then conclude this tutorial. I'd imagine that there will be some opportunity to follow up in case people have questions about this. What's your typical way of helping out? Is there some kind of an email that people can contact Lydia?
I think the user forum is a great way to start. They can start searching for the answer. They can post it, and when we post it there, other people can find the answer as well.
Yeah, so that's very helpful. So if you have any questions, please refer to the user forum. And yeah, hope this was useful.